Hello, I'm Olivia Wallace, and I'm a senior laboratory animal science student. The objective of this assignment, I chose to do the paper, and ultimately it's to understand what SARS-CoV-2 is and how it relates to other viruses, the pathology of it, and then how it relates to COVID-19 as well. And so in my essay, I just decided to answer the questions, of course. So to begin, SARS-CoV-2 is named severe acute respiratory syndrome. It is caused by coronaviruses, and coronaviruses get their name from the crown-like spike proteins that surround their exterior shell. And it results in viral respiratory illnesses, as many people are experiencing with COVID 19 also. The SARS coronavirus has a core of genetic material, which is an RNA genetic core. And many people believe that the virus originated in China from bats. That is the main theory from everyone. And then due to constant international traveling, the virus spread quickly among a whole bunch of people. Lots of people were asymptomatic as well, which caused the virus to spread even worse, which was, you know, essentially through person to person contact. And the virus travels through microscopic respiratory droplets, and that is through coughing or sneezing. And the droplets can also linger within the air, infecting surrounding individuals. It's unheard of, or there is debate on whether or how long the virus can survive on droplets that are on a surface, but it's still recommended to wash your hands and practice safety protocols. And the droplets deposit within the mucous membranes of our facial openings, so that is the mouth, nose, and eyes, whenever we inhale them or intake them in any sort of way. The infection cycle of the SARS-CoV-2 begins with the attachment and penetration. The virus will infect the ACE2 receptors of the lungs, and it uses the spike glycoproteins to attach. And the RNA virus releases into the cytoplasm, so it essentially injects itself into the cell. And it gets into the cytoplasm of the host cell. And here is where the RNA genome undergoes a translation and replication. And it said that it integrates its RNA material with the host DNA. And as a result, the virus takes over the cell and continues to make more copies of itself. And the cell has trouble recognizing that. And then as a result, the new particles burst out of the cell to infect other neighboring cells. And the tissue that the virus targets is the epithelial cells of the respiratory tract. And there is still undergoing research on just the whole pathophysiology of it all, but that is what scientists have deemed that occurs. Bats are said to be the ecological reservoir of the virus, and obviously many do favor the theory that the virus transmitted from bats to humans. There isn't, I've always wondered, but there isn't certainty on how that was, whether that was eating bats or someone were to get bit by a bat feces, or what, but they're still undergoing research on that as well. Ultimately, coronaviruses are large families of viruses that result in respiratory illnesses. COVID 19 is a new respiratory virus, and it produces a wide range of symptoms for affected individuals. Some symptoms that people have are fever, fatigue, dry cough, a loss of taste and smell, and that's typically, many people haven't really reported sneezing, but I'm sure that it is possible also. Individuals who are immunocompromised are way more likely to contract the virus, just because their immune system is going to struggle to fight it off. Let's see. The 6 feet distance rule has been enforced, obviously, and then this is due to limit the contact of the respiratory droplets between individuals. And researchers believe that the incubation period, the time that it takes to show symptoms of the virus, is going to be between 1 to 14 days. Currently, there has been vaccine rollouts, thankfully, and everyone is aware of the 3 primary vaccines: the Pfizer, Moderna, and Johnson & Johnson. And these vaccines are accessible to all individuals now. The whole goal of the vaccine is to produce antibodies, and these antibodies provide protection against future infections. When developing a test for a virus, it is important for the test to be sensitive to the virus particles. It is important for there not to be false positives. And then developing a test also is essential to limiting the spread of the virus, just so that those who test positive for the virus can quarantine so that they aren't affecting more individuals. Some things that have been done so far. Obviously, we're aware of the virtual learning and virtual communications now that have been normalized. Mask wearing, hand washing, social distancing. Although we are all tired of these measures, they are essential into returning to a normal lifestyle. And I believe that is all that I have for this presentation. And as far as this relation to microbiology, we have studied how viruses occur, how bacteria travels, and we've studied the necessary measures to inhibit these processes. And the coronavirus obviously has caused a lot of us, or researchers, to look at this on a cellular level, a microscopic level, and that is what we have done this whole time in microbiology. So thank you for watching.